Hello everybody, my name is Ian Streeter and I work at EMBL-EBI, and I'm going to give this webinar about HipSci, which is the Human-Induced Pluripotent Stem Cell Initiative. Okay, so this webinar, first of all, I'm going to give a very brief overview about iPSCs, what they are, and what is the HipSci project. Then I'm going to take you through the HipSci workflow. So this is how how the project generates iPS cell lines and then what it does with those cell lines. And then I'm going to show you HipSci's cell lines and data browser. This is at hipsci.org/lines. And this is our online resource, and this is the first place that you should come if you want to find out anything about the cell lines or the data from HipSci. And then we're going to go through some worked examples. These are kind of typical user requests when they come to use our online resource, the kind of things you might want to ask. And I'll show you how you can use our cell lines and data browser to answer some of these questions. Okay, so this is a really basic and quick summary of what are iPS cell lines, or iPSC. So this is all about the technology which takes fully differentiated adult cells, for example, these fibroblasts on the left-hand side, and we can now reprogram these adult cells by using a cocktail of transcription factors, which can reprogram them back into a fully pluripotent state. And this cell type we call an induced pluripotent stem cell. So this is still quite a new technology. It was demonstrated 11 years ago for the first time to be a viable procedure. And it's caused a lot of excitement in the field of regenerative medicine and disease modeling. It's a really exciting new tool for these fields to use. Because the iPS cell lines are generated directly from adult tissues, there's a lot of big advantages to this. So it bypasses the need to use embryos, which would be the other way of getting hold of pluripotent stem cells. Of course, there are big ethical and legal reasons why you might not want to use embryos. And also iPS cells can be patient matched, so you can take a tissue sample from a patient and reprogram that patient's cells back into an iPS cell. So this is very exciting for the fields of medicine, but there are still a lot of scientifically, there are still a lot of unanswered questions about this cell type. In particular, it's still a cellular phenotype which has not been fully characterized. And that's where the HipSci project comes in. So HipSci is a project funded by the Wellcome Trust and by the MRC. It's a collaborative project between several different institutes in the UK. And HipSci is generating a large, high-quality reference panel of human iPSCs. Currently, we've generated over 700 iPS cell lines, which are ready for banking. They've passed QC and ready to be banked. And HipSci is a global iPSC resource for the research community. So there's a big focus within HipSci to make this a global and public resource. And this applies both to the cell lines. So the cell lines themselves are available from 2 cell banks where any scientist from around the world can purchase the cell line. And this also applies to the data. So all of the characterization data and the assay data generated by HipSci are put into the public domain, where anybody can download the data and learn more about HipSci's iPS cells. Okay, so I'll show you a few slides about HipSci's workflow. So this is how we generate the cell lines and what we do with the cell lines in the project. It all starts with patient recruitment. HipSci has taken tissue samples from hundreds of healthy volunteers and also from rare disease volunteers. This is quite a kind of unique point about HipSci, that we have so many healthy volunteers in our iPS catalogue. And this is because healthy volunteers provide a powerful resource to study common genetic variation within the human population. I also have a list here of the 15 rare disease cohorts from whom we've taken, from whom we have made iPS cell lines. The next step in the workflow is isolation of the somatic cells. So this work all happens at the Sanger Institute. HipSci receives the tissue samples as either skin biopsies or blood. If it's a skin sample, then we isolate fibroblasts from the skin. And if it's a blood sample, then from the PBMCs, they get enriched for erythroblasts. The next step is iPSC reprogramming. So the somatic cells are reprogrammed using the cytotune Sendai virus, which contains the transcription factors known as the Yamanaka factors. And this triggers the reprogramming of the cells back into the induced pluripotent stem cell state. Successful colonies are then picked and cultured to obtain a sizable yield of iPS cells. The next step is quality control. So we run two different assays on the candidate iPSC lines. First of all, an expression array, a gene expression array. And with the data from the gene expression array, we run the Pluritest. So Pluritest is a calculation. It takes the data from the expression array and it looks at whether the expression profile of the cell is characteristic of an iPS cell type, and it gives it a score. So a higher number means that that cell type kind of scores highly as having an expression profile of an iPS cell type. The other assay we do at this stage is a genotyping array. And with the data from the genotyping array, we run a custom CNV check. So this is checking for copy number variations. And this gives an indication of the genetic stability of the cell lines. So we run these assays on all successful candidate cell lines from each donor, and also on the somatic cells that we use to derive it, the fibroblasts or the PBMC. From this data, we then select suitable cell lines for banking at ECAC and EBISC, two different cell banks. So the next step is cell banking. HipSci has two different agreements with these two cell banks, ECAC and EBISC. Our agreement with ECAC is that they distribute cell lines to academic researchers. A slightly smaller subset of our cell lines are going to EBISC, and EBISC can distribute cell lines to commercial entities and also to academic researchers. Our cell lines are banked to a commercial grade standard, and they must pass resuscitation and mycoplasma tests before they get banked. If you purchase cell lines from ECAC or EBISC, you do not require any further ethical approval for use in research. When purchasing a HipSci line, you will find the full terms of use within the material transfer agreement, which must be completed and signed by your host institution. Okay, simultaneously, whilst the cell line is getting banked, we also perform more characterization assays on it. So first of all, the genetic and genomic assays, this all happens at the Sanger Institute. We do exome sequencing, so this tells us what the genotypes are in the exonic regions for the cell lines. We do RNA-seq, so this gives us information about gene expression levels, but it's much richer information than the data we get from the gene expression array. It also tells us about things like alternative gene spliced transcripts. We also do methylation array. So methylation of DNA is a suppressor of gene activity. So this assay gives us kind of a profile of the gene activity of this cellular phenotype. And also for a small subset of cell lines, we do whole genome sequencing. The cell lines are also shipped to King's College in London. King's are a partner of the HipSci project. And at King's, they're doing some exciting work. They have a high content analysis phenotyping platform for these cells. And they're looking at features such as cell adhesion, morphology, and proliferation on this kind of high throughput level. They're looking at phenotypic features informative of cell behavior in an artificial microenvironment. And this is all about studying the genetic contribution on the cellular phenotype. The cell lines also get shipped up to Dundee University. Dundee are another partner of the HipSci project, and they are doing mass spectrometry proteomic measurements of the cell lines. So we've already talked about the assays at Sanger, which are looking at the transcriptome. So this is complementary data, which is looking at the proteome of these cell lines. And at Dundee, they have identified 17,000 proteins and 480,000 peptides in the HipSci iPS cell lines. So this is a really thorough and deep proteomic profiling of this cell type. And at Dundee, they have made all of their proteomics data available and visible in a tool called the Encyclopedia of Protein Dynamics. Okay, so that's the workflow of HipSci. That's what we do with the cell lines. Next, I'm going to show you HipSci's cell lines and data browser. So you can find our data browser at hipsci.org/lines. And the data browser should be, if you ever have any question about our cell lines or our data, this is a good first place to come to see if it has the information to tell you more about the cell lines and data. And this is what the browser looks like. Now, there's a lot you can do with this browser, so I'm not going to go through it all right now, but we have most of the rest of this webinar. I'm going to be giving a series of demonstrations of how you would use this cell line browser, and I'll show you some examples of how to use it, how to click around, and what you might use it for. For now, all I'm going to say is what you're looking at here is this is the front page of the cell line browser, and you're looking at a list of cell lines. So each row in this table is a different cell line, and you can see on the left-hand side of the table they all have names which start with HPSI and then the number and then a few letters. So these are the unique names of our cell lines. Okay, so the first demonstration, this is for a user who comes along and says, I want to buy a recommended cell line from a healthy donor. Okay, I'm just going to open up my web browser and share it with you. Okay, so this is my web browser. I've opened up the HipSci cell lines and data browser. This question was about just finding a recommended cell line. Because HipSci has produced so many cell lines, it can be quite daunting to select the right one. And for this reason, HipSci has selected a few cell lines to be in a recommended panel. And if you click this button at the top, then you can see our recommended panel of cell lines. We have selected these cell lines because they have high Pluritest scores. So Pluritest was the assay which looks at the gene expression profile and confirms that these have a gene expression profile which is typical of a good iPS cell line. They also have a low number of CNVs, of copy number variations. So this means that these cell lines are all genetically stable. And also these cell lines, they've all been comprehensively characterized with assay data, and they have been shown to differentiate well. So they differentiate very nicely into the different cellular lineages. So if you're new to this iPS resource and you just want a good, healthy, high quality cell line, then I recommend clicking on the button for our recommended panel. They're grouped into two different panels, one for feeder free and one for feeder dependent. The difference here is in the methods used to culture the cell lines. You might have a preference on how you would like to culture the cell lines in your lab. Now, what you could do at this stage is click on one of these purchase buttons. This would take you to the ECAC website where you could purchase the cell line directly. But what I'm going to do is I'm going to click on one of the cell line names. I'm going to click on this one called HOIC1. And this will tell me some more information about HOIC1 before I decide to buy it. I click on that. I see a cell line detail page. And there's a lot of information here. I can see that the age of the donor was in the age range 40 to 44. I can see how the cell line was derived. I can see that it comes from a female. I can see that there is a certificate of analysis available. This C of A will tell me that this cell line has passed all of the testing criteria. If I scroll down a little bit further, I can see all of the assay data which is available for this cell line. So I could download that data if I wanted to. If I scroll down a little bit further, I can see that the Pluritest has been done on this cell line, and it has a good pluripotency score. And if I scroll a little bit further, I can see that we've also done our custom copy number variation check, and this is telling me that there is very little significant kind of chromosomal rearrangements going from the fibroblast line to the iPS line. So this is a good stable cell line. So now that I'm happy that I can see this is a good quality cell line, I'm happy with it, so I might scroll back to the top and I'll click the purchase button. This will take me to ECAC's website where I can purchase this cell line. And at this point here, I would type in my credit card details and then the cell line is mine. Okay, I'm going to go back to my presentation. The next demonstration, this is for a user who comes along and says, I want to buy a cell line derived from a patient with Bardet-Biedl syndrome. Okay, I'm going to go back to my web browser again. If a user wants a certain type of cell line, or they have certain requirements, so in this case they want a Bardet-Biedl syndrome line, the best way to find a cell line that meets your requirements is to use the filters on the left-hand side of this cell line browser. So on the left-hand side, there are a whole load of buttons you can click on to filter the list in front of you. So you might want to filter by the banking availability. You might want to filter by donor consent, possibly by the assay data which is available for these cell lines. You can filter by the source material where the kind of parent somatic cell came from. In this case, we are going to try filtering for disease status. So I'm going to click this little plus, and I can see all of the different diseases which are in the HipSci catalogue currently. So I'm going to click on this filter for Bardet-Biedl syndrome. If I click on this, then the list in the table gets filtered, and it's now displaying only cell lines from patients with Bardet-Biedl syndrome. I'm going to filter my list a little bit further, actually. So I want to see only cell lines which have been banked at ECAC. So I'm going to click this button here, and I will filter the list. So it's now just displaying cell lines which were banked, which are currently banked at ECAC. So now I've restricted the list to just 49 cell lines. There are a few more things that I could do now. So by default, not all of the data is shown in this table, and there might be more things I want to know about these cell lines. So if I click on this More Columns button, then I can choose to show some more information in this table. So I might choose to click on age, so I can see the age of the donor that these cell lines come from. I might also click on method of derivation. This might be something that's important to me, how the cell line was derived. So now the table is showing me, it has an extra column with the age of the donor, and also I can see that most of these cell lines were derived using cytotune 1. Okay, so from this I might say, well, this top cell line here, this seems to have a lot of assay data currently associated with it and available. So I could either choose to purchase this cell line, or again, like before, I could click on the cell line name, which will take me through to a lot more detail about this cell line. I can see the assay data available. And when I've checked that I'm happy with it, again, I could click on the purchase button. Oh, there was one thing I forgot to show on the cell lines page. This is quite a useful thing. We have an export button here. If you click on the export button, then all of this data that are in the table in front of you you can export it and download it to your local computer as a CSV file. And when you have the CSV file, you can open this table of data. You can open it in something like Microsoft Excel or R if you use something like that. So it's quite a handy feature. Okay, I'm going to go back to my presentation. So this is just a summary. If you're searching for a particular type of cell line, go to the cell lines browser, use the filters to narrow down your search. You can use the more columns to show more information, and the export button if you want to open the list of cell lines in something like Excel. Okay, the next demonstration. This is a user who comes along and says, I want to download genotypes for open access cell lines. Okay, so first of all, I'm just going to talk about HipSci's assay data in the public archives. We have a big commitment to putting all of our assay data, all of our characterization data into the public archives so that they can be seen by the global community of research scientists. Now, because we're generating so many different data types, we send our assay data to several different data archives. Each of these archives is kind of a specialist in handling certain types of data. So this is a list of where all of the data goes. You can see that our genomic sequencing data goes to ENA, but our array-based data and RNA-seq data goes to ArrayExpress. Our proteomics data goes to PRIDE. There's an important one here, which is managed access data. And I've put that all of our managed access data goes to the European Genome-Phenome Archive, or the EGA. And it's worth talking about managed access data. and what exactly it is. So this is all to do with donor consent. So this is all to do with what consent was signed by the tissue donor when they gave the tissue sample. And the important thing here is that all of our assay data are publicly and globally available to scientists. That's the important thing. All of our data is publicly available, but some data are subject to managed access constraints. And we kind of categorize all of the HipSci data into either open access or managed access data. So first of all, the open access data. This comes from a subset of the HipSci donors who authorized the release of individually unique data to all parties. So individually unique, this means genotypes basically, genetic data. These assay data go into open access archives like the ENA and ArrayExpress. And these data can be freely downloaded by anyone. You can just go to the archive, find the data, point and click, and you have the data. It's that easy. There's no requirement to register for access. On the other hand, our managed access data, this comes from other donors who authorized the release of their individually unique data, their genotypes, for specific research use to bona fide researchers only. And this means that all of this managed access assay data, we send it to the EGA, which is a specialist archive just for managed access data. And scientists can still get this data, but scientists have to apply to HipSci's Data Access Committee before they have access. Okay, so that explains about our open and managed access data. Now I'm going to show you how you might download some open access data. I'm just going back to my web browser. Okay, so I'm back in the cell lines and data browser. I'm going to just point out that there's these 4 tabs up in the top. So far, everything we've been doing has been in the cell lines tab. We've been looking at the list of cell lines. This time I'm going to click on a different tab, which is going to open a different view. We have this files tab. So files is for looking at our data files, files of characterization data. And this shows a huge big list of all of the different data files which we have put into the data archives. So the question from our user was, I want to download genotypes for the open access cell lines. So a little bit like before, I'm going to use the filters on the left-hand side. So first of all, I'm going to filter this big long list. I'm going to filter it for open access data. So I click the open access filter button. And next, because I'm looking for genotypes, I want to look at genotypes from exome sequencing data. So in the assay filters, I'm going to click on exome seq, which will filter the list a little bit further. And if I look a bit further down, I can see that I can now filter for imputed and phased genotypes. So I'm going to click this filter button for imputed and phased genotypes. So the table in front of me is now showing only data files for genotypes from exome-seq assay data for our open access cell lines. At this point, I might decide I need some more information. I might look at the more columns button. I can see I can get the table to show what might be important. Maybe the passage number of the cell line at the point that it was assayed. I might decide that this is an important thing. And now that the table in front of me is showing the passage number, I might also decide that it's helpful to have this list of files as a list on my computer, so I could click the export button, and again I could download this list of exome-seq genotype data. I could export the list as a CSV file, which I could open in Microsoft Excel or R or anything like that. Okay, for each data file in this table, I could click on the archive button, on the link in the archive column, and this will take me out to the data archive which is hosting this data, in this case the ENA. So this link has taken me out to the exact page in the data archive which is hosting the files, the relevant files for that cell line. Each of the different assay archives has good documentation for how to download data from that assay archives. So the best thing to do is from that from the archive website, you can find good documentation with download instructions. You could also try clicking on the file download button. Now, I should warn you before you try this, some of these files can be quite large, but you know, a single file might be several gigabytes, so just be a little bit careful about downloading this data, but it should work. In my case, because I'm using Firefox, I'm going to right-click on this file download button, and I'm going to click on Save Link As. That will download this exome-seq VCF file directly to my computer. If you don't want to do it through Firefox, through your web browser, then you could also you could download it with a command line tool, or there are other FTP clients which are good at doing this kind of thing. There's FileZilla is a good example that I like to use. Okay, I'm going to go back to my presentation. So this is a summary of everything I've just said. If you want to download open access data from HipSci, come to our Cell Lines and Data browser, click on the Files tab. You can use the filters on the left-hand side to drill down to the exact data that you want. You can look out for the More Columns button to get the table to show you some more little bits of information. You can use the Export button if you want a list of the data kind of in a spreadsheet on your computer. And for downloading, either follow the instructions from the assay archives or FileZilla. I've put the link here. It's a good example of an FTP client, which is quite good for downloading large files using FTP. Okay, this is the fourth and final demonstration. This is for a user who comes along and says, I want to download genotypes for Bardet Biedl cell lines. So these are managed access cell lines. So I've already talked about managed access. These are our data which for which you have to apply for access before you're given direct access to them. Because they're managed access, if you are a user, the first thing you would have to do is apply for access. And this is done through a website hosted by the Sanger Institute. There are links to this website from the HipSci website. And this is what that website looks like. It's called the EDAM website, which stands for Electronic Data Access Mechanism. This is going to be a really very quick and brief overview of exactly how you do a data access application. There are several steps to it, and it can be, you have to make sure you go through all of the steps before you get hold of the data. So the first thing is, on the EDAM website, first of all, you register yourself as a user. So you register your email address, and you get sent an automatic email with a link to complete your registration. From that automatic email, there will be a link to click. If you follow the link in that email and click to complete your registration, now you have completed your registration as a user, but you have not actually yet done an application to see the data. You've just registered yourself as a user. So the next stage is starting an application to actually see the HipSci data. So back from the EDAM website, you can now log in as a user, find the button to start an application, and if you select HipSci from the list of agreement types, this is the start of your application. Your application is then processed. Your application will go to the Data Access Committee, who will look at your application, the reasons why you have asked for data access, and they will consider your application. Hopefully you will be successful, and once the Data Access Committee have kind of approved it, then you will have access to the data from the EGA data archive. Okay, so that's a very quick overview of the data access application process. At this point, I'm going to flip back to my web browser. Okay, because this is managed access data, Bardet-Biedl syndrome, and we won't be able to download it directly using the file download buttons. So I'm going to use a different view of the data browser. I'm going to use this fourth tab up in the top right hand corner, which says EGA datasets. If I click on the EGA datasets view, what I can see is a list of EGA dataset IDs corresponding to the different disease cohorts from HipSci. If you do a data access application for Bardet-Biedl syndrome, then you will be given access to all of the data, all of the data sets in this row here. So you will be given access to the genotyping array data, expression array data, exome-seq, RNA-seq, and methylation array data, all for Bardet-Biedl syndrome. At this point here, I can't remember what the question was. The question was that our user wanted to find out the genotyping, wanted to find the genotypes for the Bardet-Biedl syndrome cell lines. So this user would click on the link for the exome-seq. So the exome-seq assay for the Bardet-Biedl syndrome cohort, they would click on this dataset ID here. This brings up a little pop-up help menu. So first of all, this is a what are you trying to do? If you are looking for data access, then you must register for access via the Sanger Institute's EDAM website. So this is what I've just gone through. So let's assume that you have successfully done a data application for Bardet-Biedl syndrome. So now you want to download the data. And there is a link here which says, if you have been granted data access for this cohort, go to the EGA website and click to download dataset EGAD blah, 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 blah. So you can click on this dataset ID, and this takes us to the EGA website, the exact dataset that we want to download. This is the Bardet-Biedl syndrome cohort. This is exome sequencing data. And this is our most recent data release from October 2016. I'm just going to go back to my presentation. Okay, downloading data from EGA is a little, it's not complicated, but it's not completely straightforward either. This is to do with the fact that it's managed access data. But EGA, they have very good and clear documentation for exactly how to download data from EGA. This is beyond the scope of this tutorial now, but if you're trying to download data from EGA, then this download client quick guide this is the documentation you should take a look at. Okay, that pretty much concludes everything I wanted to talk about in this webinar. I should at this moment acknowledge the Wellcome Trust and the MRC who funded the whole of the HipSci project, and also I should acknowledge the HipSci consortium, which is the 4 institutes listed here, and also all of the clinics who donated tissue samples.